So before I start, I just want to thank the funding agencies that have funded a lot of the work that I've done, and this includes the George Melendez Wright Climate Change Research Fellowship from the National Park, also the Southern California Research and Learning Center that's partnered with Santa Monica Mountains Fund, Seabird Electronics, who has loaned me oxygen sensors, which has been very helpful. The Bureau of Ocean Energy Management has purchased a pH sensor that we now use in the national park, and then also the National Science Foundation. So I am a graduate student in Gretchen Hoffman's lab, and the Hoffman lab really is a lab that studies physiology. And we do usually large-scale physiology studies. So that means we go to different environments, maybe within the same species, they're across their entire range, or we go to different biomes like the tropics, the Antarctica, for example, but also we study a lot of organisms in our backyard here. So generally what we are interested in is how organisms are responding to their current environment and also how they might respond to future conditions. So the way we do that is we go into the environment, we measure the environment, whether it be pH or temperature, salinity. We collect organisms from that same site, so we know what their exposure is to all these different parameters. We set up experiments, usually in the lab, but we can also do this in the field. And this is a photo of sea urchin larval cultures. And then we measure some type of physiology, like respiration rate or gene expression, fertilization rate of sea urchins, for example, or we can study growth of organisms. And we do this to try to get an idea of how animals are responding to present-day conditions, but also how they might respond to the future. And so the things that I want to talk to you today about are listed here. And I hope to answer these 5 questions for you. So ocean change encompasses a lot of different processes, including, for example, warming. But I'm going to be talking about ocean acidification specifically first. And then I hope that you'll be able to see why we should care about this and what the current conditions are like in the Channel Islands and what the next steps are in research, and finally, what you can do about it. So I'll start off with what is ocean acidification. So please raise your hand if you have heard of it. Oh, it's almost everybody. And okay, keep your hand up if you feel like you understand it really well. A lot of them went down. Okay, keep your hand up if you feel like you've explained it successfully to someone else. A few. Okay, good. Okay, well, hopefully today you'll have a better understanding of it, and hopefully you'll be able to share your knowledge. So I'm going to start with a demonstration. So I'm going to need 3 volunteers. In science, come on up. In science, we do everything in triplicate at least. Sure, one more. There we go. Okay. So I'm going to give you each 2 cups. One will be the control, and the other one will be the treatment. And I brought seawater with me from Santa Barbara. Better than filtered. That's what. It's actually from the lab, which means it's very, very filtered and very clean. Oh, good. So it's not from the harbor. Yeah, it was warm because it was in my car for a while. Here we go. Okay. So I have 3 straws. I guess I should have. You do not have to drink the seawater. So through this straw, you guys will just blow air and bubble it into the seawater. And we'll do this for, I used to do this myself, but then with talking, I got a little out of breath, so now I recruit volunteers. So go ahead and start. We'll do this for a minute or so and just bubble up, but don't move the straw to your control cup. So who can tell me what's happening? What you remember about ocean acidification? Sorry? Adding oxygen, yeah, a little. Carbon dioxide. A lot of carbon dioxide is being added. And who might know what that's doing to seawater? It's changing the pH. You guys are so knowledgeable. So we'll go for 10 seconds more. Give it all you got. So I have bromothiamyl blue, which is an indicator dye. And the color scheme is up here. So the seawater that's blue has high pH, and the seawater that has anything on the scale of yellow or green is lower. All right, very good. Okay, so I'm going to add some drops to these. And you can use your straw to stir it around. Oh, sorry, I'll do it since your hands are full. There we go. And if you guys could walk around the audience and just show the difference.
Is this like a breathalyzer thing?
Yeah, it is. I will not be walking around.
It only measures pH. There we go. So the only difference is that we've added carbon dioxide.
And it reacts that quickly.
Yeah, the reaction can be really fast. But a human breath has a lot of carbon dioxide, I think around 40,000 parts per million. So we're adding a lot of carbon dioxide, a lot. And so when CO2 mixes with water molecules, it creates carbonic acid, and that's why we see the change in pH. And so this is actually the process of ocean acidification that you guys have just witnessed. Please thank our volunteers. Thank you. You can leave the cups on the floor. Yeah, that'd be great. I'll grab them later.
Yes,
Yeah, we can see how fast it equilibrates with the atmosphere. Okay, so ocean acidification, which is this decline in pH, has been documented really well. And so this is some data taken by NOAA at Hawaii. So this red curve is the increase in carbon dioxide. So this is from 1960 till about 5 years ago. And carbon dioxide concentrations in parts per million are on the y-axis. And you can see this steady increase in the red line. The blue line underneath it here is the increase in partial pressure of carbon dioxide in the seawater. And so you can see that the rate of that increase is very similar to what's happening in the atmosphere. And this corresponds to a decline in pH. And so it's this decline in pH that's called ocean acidification. Not because the ocean will be acidic, because acidic solutions have a pH less than 7. The ocean pH, you can see, is above 8.0 here. But it's the trend, so it's the decline in pH why we have this term ocean acidification. And the reason for this is, of course, burning of fossil fuels releases tons and tons of carbon dioxide into the atmosphere each year. And there's a small component where deforestation actually causes a reduction in the CO2 removal from the atmosphere. So, but this component is much, much smaller, of course, than what we're burning for fossil fuels. And then these molecules equilibrate with surface layer of the ocean, mix with water molecules, and that's caused the decline in pH. So based on predictions of carbon dioxide emissions, with various scenarios, say we're doing some climate mitigation or we're increasing industrial production, we can make predictions about carbon dioxide concentrations in the atmosphere. That's in this top panel here, from 2000 till the end of this century. And you can see some of the worst case scenarios are nearing 1,000 parts per million. This causes a decline in ocean pH that we can also predict. And so based on these different scenarios, we might expect pH conditions to be around 7.75 for the end of the century, depending, of course, ultimately on how much carbon dioxide we emit into the atmosphere. So the first answer to the question, what is ocean acidification? Ocean acidification is the increase in seawater acidity, or the decline in pH, that's due to human-induced carbon emissions. So moving on, why should you care about this? I'm going to go through 3 examples of organismal responses to low pH. And the reason that we want to find out how organisms respond ultimately is because we are interested in how ecosystems function. A lot of nations rely on seafood. We eat a lot in California as well. And so it's also a food security issue. So the first example are pteropods. So pteropods are a free-swimming marine snail. They have a shell just like a land snail does, but they, instead of their foot, they have almost like wings, and that's how they swim around in the water. And they're at the base of the food web, so anywhere from krill to whales eat these little guys. They're coined the potato chip of the sea because everything eats them. And they're found in all places. So they're found off of our coast, but they're also found in the Southern Ocean and Antarctica. And they're very, very important because they're a food source for many organisms. And if you take one of these shells and you put them into future pH conditions, so what's expected for the end of the century, this is a progression of what that shell looks like. You can see that it starts to deteriorate. So what's actually happening is that this shell is dissolving very slowly. And this, of course, is problematic. And some of this shell dissolution has already been documented in the Southern Ocean. The next example are sea urchin larvae. So these look nothing like sea urchins, but this is what a baby sea urchin looks like. It's free swimming also, and it has a little skeleton inside its tissue. And this skeleton is also made up of calcium carbonate. And if this is one of the earliest ocean acidification studies, so these treatments that these larvae were grown in went all the way down to, I believe, a pH of 6. So that's not what we would expect for the future. But you can see that there's a trend in declining size. And this has been found in many, many species of sea urchins, especially, or also the ones off of our coast here. And the change in size for what's considered present-day ocean versus end of the century is about 10%. And so this can have really big cascading effects if this is happening to all larvae in the ocean, and it's reducing their survival. And so what's happening with calcium carbonate, why it dissolves, it's like the name is, calcium and carbonate ion. And when you put calcium carbonate into an acidic solution, the calcium carbonate dissolves. So this is the same process if you have ever taken Tums. Raise your hand if you've taken Tums. A lot of people, yeah. So if you feel nauseous, it might be because your stomach is really empty and you have a lot of extra acid in your stomach. If you look on the back of a Tums box, you can see that the ingredient is actually calcium carbonate. So it's kind of like eating a little shell. And so you take that, the tablet dissolves and neutralizes the stomach acid. So that's, in effect, what ocean acidification is doing on a very, 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 very slow scale, and definitely not as intense as stomach acid, obviously. So the third example are from oyster larvae. And so these are photos of one-day-old oyster larvae from the Taylor Shellfish Hatchery in Washington. And these shells obviously look different. One is very smooth and very large. The other one has some undulations, and it's much smaller, and it looks a little bit irregularly shaped. And so at one point, these, you may have heard this in the news, the oyster farmers in the Pacific Northwest were having a lot of crashes. So they were using a lot of yield, and this had a real economic impact on these businesses. And what was happening was that periodically, lower pH seawater was brought into their hatcheries, and the oysters couldn't handle it. So they couldn't build a strong shell, and it caused their whole cohorts to get wiped out. So they partnered with scientists to try to figure out what was going on, and they figured out that it's because of this sort of lower pH water that's being brought in. So now they just shut off the water flow during those events and then keep the high pH water. So there's some management involved. You can't see the credit here, but these photos are from scientists at Oregon State. So why do we care about ocean acidification? Ocean acidification makes it difficult for organisms to build a strong shell. I put an asterisk there because this is the finding that a lot of the early studies have found, and a lot of species that have calcium carbonate exhibit this response, where the growth is stunted under these low pH conditions. However, there's a lot of differences between species. Some species actually will perform better, and some species have no effect. So scientists are really trying to figure out what makes an organism sensitive and what makes it not sensitive. And so we'll get to that in a little bit. So what drives the current conditions? So these projections of ocean pH are based largely off of processes that occur on a sort of open ocean scale. And they're the projections that I showed you earlier of about 7.7 or so for the end of the century is what we use to create future conditions in the lab and to expose these organisms to and see how they respond. But is that really representative of nearshore conditions? So is the open ocean the same pH as coral reefs or in kelp forest or in coastal Antarctica? So I've become really interested in characterizing these natural pH dynamics to better understand what organisms are actually experiencing. So this is data that we've collected from Moorea, which is a tropical coral reef, coastal Antarctica, and then right here in our own backyard, the Santa Barbara Channel and in the Channel Islands National Park. And what you can see, first of all, is that the traces all look different. So pH here is on the y-axis. It's the same scale for all three graphs. And then time is on the x-axis. So the length of these time series data are a little bit different for each plot, but the increments are by each week. So that's how you can compare. So you can see that, for example, even within Moorea, if you are an organism living on the fore reef, you see this very small daily cycle. But if you're living on the fringing reef, so the reef that's closest to the island, you see these larger cycles in pH. If you are an urchin in Antarctica during the spring or during the early summertime, you see almost no change in pH. And if you are an urchin in California, you might see a really wide variety of pH. And remember that the future predictions in the worst case scenario were between 7.8 and 7.7. So you can see that maybe those levels are not really representative of what animals will experience in the future if they're already experiencing them now. So we can zoom in a little bit more, and we'll do this with data from the Channel Islands. So I started working with Park 3 years ago, and we set up the first pH sensors on Anacapa Island and Santa Cruz Island. And then later we moved the Santa Cruz sensor to San Miguel. And these are all really different habitats, so we can't really make obvious direct comparisons. Anacapa Island is inside a kelp forest. It's on the pier at the landing cove, which you guys may have been to. Sometimes on a very clear day, you can see it. We wrap it in really bright yellow electrical tape. So sometimes there'll be if you see a glistening of yellow, it might be the pH sensor. The sensor at Santa Cruz Island is also on the pier at Prisoners, and this is in the middle of a seagrass or an eelgrass bed. And then the sensors at San Miguel Island are actually a little bit offshore, so it's in deeper water of 60 feet, and it's not inside kelp. And so here are the data for San Miguel Island in red and Anacapa Island in blue. And here are photos of those two sites. So here's the sensor at Anacapa. It's a bit dark, but this is the pier piling here, and then some kelp. And then here's the sensor sort of in open water at San Miguel Island. And what you can see is that the curves do not look the same. And so the y-axis goes from 7.8 to 8.2, and this time series for the red line is exactly 1 year. So we have a little bit more data for Anacapa. So let's zoom in to this to see why they're different. And so if we look at Anacapa Island pH close up, pH here is on the y-axis from 7.9 to 8.2, and it's this blue line. And you can see this pattern of the pH going up and down, up and down. And if we plot that next to oxygen, so oxygen is the pink line with the y-axis here, oxygen saturation, we see a very similar pattern. So who might know why that is? Yes. I heard that. And what were you going to say? The photosynthesis, exactly. So when kelp is photosynthesizing during the day, it's taking up CO2 from the water column. And it takes up so much that it actually causes a change in pH. And it produces oxygen. So remember, if CO2 is removed, you're taking carbon dioxide out of the water. The opposite of ocean acidification, the pH goes up. Exactly. Yeah. And then at night, oxygen is used, carbon dioxide is produced. So that's along with the trend of acidification, so that causes pH to go down. So we get these really clear daily cycles in pH. And so that just means that biological processes can have a really strong impact on pH variability. And this could affect the organisms that are living in the kelp forest. So if we look at Santa Cruz Island, so this was data from an eelgrass bed. This is now a pH plot with Santa Cruz Island pH data in pink and Anacapa in blue. And you can see, again, that there's these really clear daily cycles. So this is a little over a month-long data series. And if we look at for every observation from Santa Cruz Island, and we plotted that pH observation along with the corresponding oxygen observation, we see this really clear linear relationship. And what that means is that oxygen variation explains a lot of the pH variability. So in fact, this value here, a little bit more than 70% of the pH variability that we see can be explained by the variability we see in oxygen, which just means that the process that's driving both is really connected. It's the same process, and that is photosynthesis. And so as you expect that with kelp, that also happens with the eelgrass. Okay, so now we can look at San Miguel. So I've zoomed into a period in the spring. And if we take all those pH observations in the red line and we plot the salinity that was measured at that same time against the temperature, we get this salinity temperature plot, which is all these dots. And every dot is color-coded for the pH value that it has. So these are all different moments in time for that one period in spring. And what you can see is that there's blue dots at high salinity and low temperature. And blue dots mean low pH. And then the red dots mean high pH. So you can see that the low pH at San Miguel Island is occurring when the water is salty and when it's cold. And who might know what that process could be from? Upwelling. You guys are very good. Exactly. So upwelling of deep water will cause this trend to appear. And so when the winds are very strong in the spring, they blow south. And with the rotation of the Earth, that causes the surface layer of the ocean to move offshore. And when that happens, of course, there's water that has to replace that water, and that comes from the depths. And that water is old and it's cold. So it's old, that means that it has absorbed a lot of CO2 in its water parcel lifetime, because in the deep ocean, there is no photosynthesis. So only the organisms that are breathing will be contributing CO2. So that causes low pH in these deeper waters. And it's cold because it's been really isolated. So that's why when we get these cold temperatures at higher salinity, water is more dense, and low pH, we know that this is indicative of an upwelling event. And so oceanographic processes can also contribute to pH variability. And so to understand the current conditions, even at different sites, even within the Channel Islands, which is a pretty small spatial scale if you're thinking in terms of processes that are much larger, it's really biological and oceanographic processes that drive the pH observations that we see. And so if you're an organism, say a sea urchin species that lives in all of California, that doesn't mean that every population experiences the same pH. And we'll get to that, why that's important next. So what are the next steps now that we know that there's this natural pH variability that exists? So these are, I'm borrowing examples from other people because they're very good studies. This is an experiment done by Christina Frieder when she was a graduate student at University of San Diego at Scripps. And she exposed mussel larvae. So she was growing baby mussels in different conditions. One condition was high pH for 8 days. The second condition was low pH for 8 days. And the third condition was low pH, but she included this daily cycle in pH variability. And then she measured growth. So here is shell length on the y-axis, and here are the three conditions in the same order as shown above. And what you can see is that at high pH, the shells are larger, and at low pH, the shells are smaller. And that's really the same trend that I talked to you about with pteropods, with sea urchins, and with oysters. And this is for Mytilus californianus, which is found off our coast. She did this with another species, Mytilus galliprovincialis, which you can also find. And the same thing at high pH, the shells they looked very normal, they're large. At low pH, it declined, but there's still quite a bit of variability in shell size. But then the remarkable part is that when the larvae were exposed to that varying pH, even though it was low, their shell size was still high. So what this means is that some species could actually benefit from being exposed to this natural variability in pH. So the answer is that variability can matter. And so that's in terms of like these daily cycles that are caused by kelp forests and eelgrass beds like I showed. The second example has more to do with upwelling. So this is a study that was done by Dr. Morgan Kelly, and she was a postdoc in our lab, and she looked at sea urchin larval development. And she collected urchins from two sites, one in Southern California, where upwelling events are less strong, and one from Northern California. And upwelling in Northern California is much stronger. So what would that indicate? Lower pH. More frequent exposure to low pH. And so maybe the urchins from these northern sites, because they already experience low pH, maybe they're a little bit more tolerant. So she crossed a lot of urchins together, males and females, and she measured growth of the urchin larvae. And this is the trend she found for the the length of the larvae for all the larvae whose father came from that site in Southern California. And you can see the larvae is large at high pH and is smaller at low pH. And that difference is about 9%, which is what is commonly found. But when she did this with larvae whose father were from Northern California, she saw the same trend, but it was less strong. So you can see that while the sizes of the larvae with fathers from Northern California were still smaller at low pH, they weren't as small as when their father was from Southern California. So what this means is that it's possible that there are some populations that are locally adapted. So sites that experience really low pH could become very important for maintaining genetic diversity, to try to maintain these individuals who can withstand the conditions. So the trend is still declining. It's not a complete rescue of size like we saw with the mussels, but the difference is less great. And so what does this mean in terms of marine protected areas? Marine protected areas, if they are in locations of kelp forests or eelgrass beds, places where there's a lot of photosynthesizing organisms, or species, I should say, these could become really important in terms of ocean acidification. So marine protected areas are set up to try to conserve the natural resources and maintain healthy populations. But they could also function as a refuge if natural variability ends up being really critical for certain species. The other thing that marine protected areas can aid in is if they're very large enough such that they can support large populations of, for example, sea urchins. In large populations, you have more genetic diversity, so you have more of a chance that some individuals will actually perform well in the future conditions. And just maintaining these healthy populations is probably our best bet to try to make sure that organisms will withstand these changes. So maintaining these large and genetically diverse populations maximizes its evolutionary potential that Morgan saw in her study. So the update about what's the next step is really to try to understand better which species are sensitive to what. So we see a lot of variability in organisms' response to low pH, but again, these are all stable conditions. When we see in nature, there's actually quite a lot of variability depending on where in the world you are. And then, of course, trying to understand if there's potential for evolution. So are species able to adapt fast enough such that they can overcome some of these hurdles? So lastly, I want to leave you with a slide about what you can do. Because these global processes can sort of be overwhelming to think about what you as an individual can do. I have brought you some ideas. So the first thing is to educate yourself, which you're all doing by attending this lecture or watching online. So that's really great. Sometimes ocean acidification is in the news. And you can also find a lot of great resources online, especially at NOAA. NOAA's website has really good resources. And then the next step would be to share your knowledge. It's incredible how many people don't know about ocean acidification. I'm really happy to see that all of you guys knew about it. So making sure that other people know about it is really important, but also being aware. I think one part of that is being aware of what's currently going on. So that's when I talk to audiences like you guys, I always try to describe sort of the process and where we are in scientific research, because we don't have all the answers yet either. Always you can support ocean conservation efforts. Maintaining the healthiest populations is probably our best bet. And vote. You can vote. Vote for things or people, measures that share your views. For example, for reducing CO2 emissions, protecting ocean resources, supporting science funding to continue this work. And then obviously, you can always try to reduce your own carbon footprint. And with that, I'd like to thank you for listening, and I'll be happy to take any questions.
If anybody has any questions, raise your hand. I'll come over with the mic.
Hi, I have two questions.
Yeah.
First question I have is, what is sea-friendly transportation?
Carbon-friendly transportation, sorry.
And then the second question is, I was wondering, we've learned that there are a lot of emissions in the channel from the natural oil seeps.
Yeah.
And I'm wondering, does that have any effect, or has anybody studied that effect on organisms?
So you're talking about the methane
Right.
bubbles, or just the oil, like the raw
methane
oil?
bubbles, I guess.
Yeah. So they do not mix with water in the same way that carbon dioxide does.
Okay.
Yeah. But people are studying those.
Any others?
lot of talk about fracking and the impacts in Ventura right now. A lot of dumping going on. I don't know what the facts are. Any information on that? On the sea life?
I know very little actually about the resulting effects of that kind of runoff, and yeah, I'm not sure. Depending though on what kind of chemicals are in there. For example, if there's material in that water that actually, for example, provides nutrients to photosynthesizing species, you can get more phytoplankton blooms, which could really change pH dynamics. Of course, if those chemicals are killing a lot of organisms, then you'd also presumably see that in the pH variability. Yeah, but I'm sorry, I don't know the exact one.
I'll come over there.
Hi, and in the future research that you said it's ongoing.
Mm-hmm.
So what can we share? What are some focuses on different species that you know of that are going to be coming up in the research that will be coming?
Well, when I mean ongoing research, it's like, for me personally, that's what what's ideal. So I would be really interested in understanding the physiological mechanisms that will allow some species to perform better during this variability exposure. Like, is that just because they have the right genes, or is that because actually those organisms in the wild have experienced that before and that's why they're tolerant? Really, the adaptation is something that I think the whole scientific community struggles with. It's really labor intensive to do these experiments, and it's even more labor intensive to try to generate these cycles in the lab. And so because a lot of the species that we're interested in, like sea urchins, have actually quite long generation times, it's really difficult to do these multi-generational studies. So people like Morgan Kelly go about that by looking at heritability from populations that have spatially experienced different things. So that kind of work is still ongoing. There's a lot of work that's still just expanding into other species. So while we have exponentially increased our knowledge about what species respond to what conditions, it's a very small proportion. So people are expanding now more to fish and sharks and things.
And the second part, how much cooperation between countries, different areas, is this research that we're seeing coming up? Is there much cooperation with other countries?
Yes, there's a lot of international collaborations just within this whole field. Certain places in the world create an environment to ask different questions. And so our lab partners, for example, with people in New Zealand to do work in Antarctica, for example. And there's a lot of, there's a lot of international collaborations. It's really a global effort. I mean, this is happening everywhere in the world, and it's been really well documented. So every nation that has some sort of dependence on the ocean should really be thinking about this. And actually, I have Lois Capps on here because she just recently introduced a bill and allocated funding for scientists to partner with aquaculture facilities to try to create collaborations that are, outside of academia and really aim to mitigate what's going on or understand what's, work locally.
I was wondering if there's any studies that you know of that looked at what the natural pH is, like say 500 years ago. If you have any kind of, so to see if there's any kind of natural trend.
Yes. So obviously there have been periods in time where there's been a lot more CO2 in the atmosphere, and it's really difficult to compare those events directly to what we see now because well, we don't have the same instruments, obviously, as millions of years ago. And, but the rate is really different. So they've been able to document CO2 concentrations in air for a really long time using ice cores and things. And the rate of emissions now is beyond anything that they think has occurred in the past 300 million years. So that rate is one part of it. The other part is that, I didn't put this up there, but pre-industrial era, global average ocean pH was considered to be 8.2. And so since then, so that was like 1750. Since then, the pH has declined by 0.1 unit. And that sounds like a really small amount, but that's a 30% increase in acidity. And so now mean global ocean pH is considered to be 8.1. And then that's predicted to now decline anywhere between like 0.3 and 0.4 units, but within 100 years. And so that's a really, really big difference. And there really is no good analog for that. The acidification events during in the fossil record have occurred during periods where a lot of organisms have also gone extinct. And so, but it's hard to compare those events, but we know enough to be concerned. So.
Back to the lowest caps bill. One, what's the status of it, and does it have a chance of passing?
I'm not sure. I really hope that it does. That would be great.
What started your interest in this topic?
It was a bit by chance, actually. So when I was an undergrad at Oregon State, I was always interested in marine biology. And I ended up doing some lab work that I thought was so tedious that I was like, no, I can't, lab experiments are just not for me. So I decided to do an internship, and I found one in Australia that was supposed to be this really big dive project. And so I was like, great, I'll be out in the ocean a lot, and I'll be doing a lot of diving. And I got there, and the project kind of fell through, and I ended up doing lab work. But the lab work this time involved a little bit of snorkeling, collecting urchins, but it was actually just a project on ocean acidification and early sea urchin development. So this was back in 2008. And I just got really interested in the topic. I've always been really interested in how humans interact with the natural world, and how our relationship changes with it. And so the ocean acidification I kind of stumbled upon, really enjoyed the process, so both fieldwork and the lab work that came with it. And it kind of went from there. And I came back to Oregon State and joined that same lab and did a lot of lab work, but enjoyed it again because I had a greater perspective about what kind of work goes into generating real conclusions. Like every graph you see, there's so much work behind it. It's incredible. It's hard to know, but yeah, so you gotta love the nitty-gritty also.
In terms of in terms of marine protected areas and using them as a way to preserve genetic variability, is it better to have a few large areas or a network of many, many smaller protected areas?
I think it depends on the goal. So if you need to have small pockets of refuges, say you have a certain amount of square miles that you can either divvy up or keep together. If you divvy it up along the coast, maybe you are providing protection for organisms that travel really far and go really far between. If you are really concerned about a species that doesn't go very far or whose range is really small, those might benefit from just a single larger one. So it kind of, it's a very good point, and I think that's a really challenging topic. But I think it depends on what process you're targeting, I think. Probably a combination.
And connected to that, I mean, you talk about the photosynthesis and how big kelp is in creating this, relieving the problem.
Well, maybe. That's
Maybe.
we're in the very beginning stages, yeah.
But I mean, then the marine protected areas in reference to kelp becomes another possibility.
Exactly. So it's possible that marine protected areas can become really important in terms of supporting organisms and their ability to deal with changing ocean conditions. Yes?
Your studies now have been on small animals and organisms. Is the goal to get up the food chain to larger mammals and do a baseline study now of what's going on so you can find out what's going to affect them in the future?
Yeah, so that's a very good question. Well, it's much harder to put a mammal inside the lab at the amount of replication that's required. I think those effects are really going to be seen through cascading effects, so changes at the bottom of the food web interacting such that maybe food supplies are changing, becoming more abundant or less abundant for the marine mammal of interest, for example. The real goal, like the next step for this research really is to understand ecosystem level processes. So just like you said, like moving up the food chain. So if one, let's say, sea urchin responds a certain way, but then the, what's that fish? A sheephead who eats a sea urchin responds a different way, or maybe their interaction is different, such that the urchin's response to ocean acidification becomes sort of meaningless or not. So those are interactions between species. People are really trying to look at that now. And the more you can, the more interactions you can test, the better able you can scale up to this ecosystem level. But it's incredibly difficult. And so there are experiments now that are ongoing in Antarctica. There's one in the Mediterranean. There's one up north in California where they've actually deployed, they're sort of like aquariums that are open on the top, and they're acidifying the water inside of them so that they can look at, in the ocean under naturally varying pH, temperature and light, they can locally acidify like a small reef or like a small patch of a seagrass bed. And so that's like one way they're trying to get at these bigger level questions like ecosystem effects, because ultimately that's what we're interested in. And then obviously fisheries and aquaculture, which are single species, so that might be like a little bit more straightforward to study. Yeah.
Did you look at temperature variability in your?
Yes. Yeah. So I did. I didn't show it here. Temperature does impact pH, and there's seasonal warming that occurs, but that analysis is like still kind of ongoing. Yeah. The ocean is getting warmer, and as the ocean's getting warmer, that causes a whole other suite of processes to occur, and how that interacts with ocean acidification is really challenging to predict. So, for example, with warming, waters are more likely to stratify, in which case the turnover between deep and surface layers is going to be less. And so if, for example, there's organisms that are taking CO2 out of the water for months at a time, for example, during the Antarctic summer, because it's 24 hours daylight, the pH actually goes really high up. But if that water isn't replenished, those organisms will run out of nutrients. And so stratification can change pH dynamics because of the indirect effects of warming on biological processes as well. So making these interactive predictions is really challenging. But that's a very good question, because ultimately all these things are happening at the same time.
What kind of a correlation is there made between the rainforest ecosystem and the ocean ecosystems and how they might play a part together in this acidification?
Let me think about that.
You did mention something about deforestation.
Okay,
earlier,
yeah.
and, you know.
So the deforestation, what happens there is it's like we're removing a lot of kelp from land, essentially. So except that forests actually store carbon. So we've removed some amount of biomass that's actually able to take up carbon and store it for a very long period of time. And so while, say, planting a tree, or, you know, you would be helping absorb carbon dioxide a little bit, it just doesn't compare to the fossil fuel emissions, but it's enough of an effect that, you know, that it's scientifically relevant. So deforestation just hasn't helped, but stopping deforestation won't solve the problem by any means.
Hold on just a minute.
I just happen to think about the Santa Ana winds. Do they have any short-term effect when they blow about all that carbon going in the water?
I don't know that directly, but winds in and of itself definitely have an effect. So the direction of the wind in terms of the movement of the earth can cause upwelling events, or it can cause downwelling events. Like, we don't see very many upwelling events at Anacapa, for example, because the winds are running in the opposite direction of what would cause upwelling. And the upwelling that we see locally really occurs a lot on the north side of the channel. So if I go back, here, this blue line is from a site at the mainland where upwelling is a little more prominent, whereas the pink site here is at Anacapa Island, where we don't see that upwelling. So that's how wind really impacts the pH variability. Wind can also cause just water turnover, and like I said, like bringing new CO2 up. And in Antarctica, for example, where phytoplankton blooms take out a lot of CO2 in the water, that mixing of the surface layer with deeper water brings the pH back down.
Any other questions? Okay. Thank you, Lydia.
That was lovely. Thank you. Thank you.